Hello everyone. Now we are going to discuss about the SAR of local anesthetics. Before going to the topic, we have to know what is local anesthetics. So local anesthetics are agents which produce reversible loss of sensation. So when we are applying the local anesthetics in a particular area, that will cause a loss of sensation. Okay, there is no sensation in that particular area. Let us see the SAR of this local anesthetics. See, most of the local anesthetics, they consist of three parts. One is lipophilic center, intermediate alkyl chain, and hydrophilic center. So lipophilic means they are having affinity towards the lipids, whereas hydrophilic will be they are having the affinity towards the water molecule. Okay, so here we will find the structure of this one of the local anesthetics. It is having the basic nucleus like this. Okay, so aromatic ring will be present, then it is followed by the alkyl chain, or it may be a ester or amine. Okay, then CONH2 like that it will present, or COO like this it will be present. Okay, this is followed by that it will be attached to this alkyl chain or ester or amine, that will be attached to the tertiary amine. Some other substitutions will be there. So it is a hydrophilic center, and this aromatic ring is lipophilic center, and here it will be attached by the alkyl chain. Okay, this is the main parts of the local anesthetics. So apart from these, this lipophilic center, which is responsible for the lipid solubility, that is essential for the anesthetic activity. So if the compound is having the lipophilic activity, means it is responsible for the anesthetic activity. So it plays an important role since their action depends upon their ability to penetrate the cell membrane. So first thing we have to know that lipophilicity is an important factor. Why it is important? Because of their ability to penetrate the cell membrane. Then lipophilic center is either carboxylic acid or heterocyclic ring. So we have seen that one. It may be an aromatic ring that has been attached to the carboxylic acid, or it may be a heterocyclic ring. Okay, that is responsible for the lipophilicity. So it is responsible for the local anesthetic activity. Here we have taken one example, that is hexylkyne. See, this is the structure of hexylkyne. Here aromatic ring that has been attached to the carboxylic acid, and the CH3, then CH2NH, cyclohexane ring will be attached. Okay, so the next point is hydrophilic group. The hydrophilicity. The hydrophilic group can be either in the form of secondary or tertiary amine, or part of a nitrogen heterocycle. See, we have seen lipophilic center, hydrophilic center, and it will be containing an alkyl chain. So this nitrogen amine, that is tertiary amine, that is the hydrophile that is present in the local anesthetics. Here we have seen that one of the example, that is Procaine. This is the structure of Procaine. Here NN diethyl amino group will be present. So here para amino benzoate will be present, that has been attached to the alkyl chain, that is CH2, CH2, then N C2H5, C2H5. This diethyl amino group, that is the hydrophile that is present in this nucleus. Then the presence of piperidino or pyrrolidino group as hydrophilic center gives the used to identical activity. If it has been substituted with the any heterocyclic compound such as piperidine or pyrrolidine. So piperidine, it is a 6-member ring with one nitrogen atom. Here we have seen one structure, that is piprocaine. This is the structure of piprocaine. Here benzene that has been attached to COO, CH2, CH2, CH2. Here 2-methyl piperidine has been attached. This is the 6-member ring. To this nitrogen is attached. In second position, methyl group is attached. So this is the 2-methyl piperidine that has been attached to the alkyl chain. So presence of any heterocyclic compound such as piperidine or pyridine, that is also responsible for the hydrophilic ring. Then the linkage between the aromatic ring and the amino group is either ester or amine. So here we will find this one. Okay, so we have seen aromatic ring and tertiary amine that will be connected by the any linkage such as ester or amine. It may be RCOO or RCONH2. So they may be either short hydrocarbon chain or oxygen or nitrogen or sulfur atom. So here we will find the structure of lignocaine. This is the structure of xylocaine or lignocaine. To this xylene has been attached and diethyl amine is present. This has been attached to the amine. That is here you will find that one, CH2CONH. Okay, so the amine or ester linkage will be present in that one. That is responsible for the local anesthetics. So this is the SAR of local anesthetics. So we have the points we have to remember is lipophilic center, intermediate alkyl chain and hydrophilic center. So lipophilic means that aromatic or heterocyclic ring, and hydrophilic ring will be tertiary amine that has been attached to the alkyl. These two are attached by the alkyl chain, ester or amine group. So I hope you could understand the SAR of local anesthetics. Thank you all. Thank you.